还是信号不太好，还是怎么着？哎，刘老师，刘老师啊，
不行啊。
对你那
哦，
边声音我们听不到，
那我听得见你们呢，我也看得见我自己啊。
你在那边说话，我们在线上听不到。您要不试下看手机的热点？你手机有网络吗？
手机
嗯，
热点怎么上呢？
就是手机就在移动通讯里面可以打开热点，然后就相当于把自己的手机当成这个路由器，就
我这个网
在设置。
络没问题啊，这个我看得见你们呢。
啊，
现在可以，
现在能听到了，
现在可以了，
现在能听
现在可以。
到了。嗯，
刚才有点，
刚才一直听不到声音。
我一直看得见你们那个，也能
那要不
什么？
您
要不然
share 一下 screen 试试看？
好吧，我共享
嗯，
一下。
现在能听到。
可以吗？
暂时还没
嗯，
出来，
看不到。
稍等一会儿。然后你在那个房间里面有没有那个网线的 cable
哎呦，没有哎。
啊？那不过有的时候它那个笔记本电脑可能也不一定有端口，就还是只能用它的无线网。
就是
还没
一直停留
出来吗？
在，
还没有。
没有。
我这已经共享了，可以的。胸镜你也看不见。
对，
看不到，
我看不见。
一直显示一个黑屏，上面写着 has started screen sharing，然后就没有的。
哦，可以了，
然后出来
稍微有
了，
点慢。
可能就是比较慢。
哇哦，希望不要眼睫膜。如果我
OK
掉了线，我在这讲，
能听到，现在能
你就给我
能听到。
打电话。
好，
OK，yeah。
要是讲的时候，要是
看看，
摄像头关了，可能会好一点。就
我的
是
摄像头吗？
如果本来就慢的话，
对，要不
对，
把你的摄像头关了，
就是讲
听到
的时候吧，就是现在可能刚开
对，
始介绍，
现
大家还是
在对，
想看一看。
那我现在已经关不了了，没有窗口让我关了，我已经共享了。
对现在
现在这个网
现在都，
速还行，没问题。
嗯
我们可以再等一会儿再开始。
嗯，行，再过2分钟，还没有到时间呢。咱们的 panelist 都到了啊，叶伟到了吗？
当然。
啊，
哦，我才知道我这个在这个，
叶伟好像还没有看到啊，你没有开摄像
他
头可能
说话了。
看到了，看到了，看到了。
我在我等一下，我不知道这个，我想把背景 filter 一下，现在在这个，
嗯，对
但我听着了，我也能看得到这个 screen。
对。
好的，
我把
好的。
大家 panelist 设为 co-host，他们就会在那个 list 的前面。
嗯，那我们现在开始吗？还是
可以啊，就我们一边开始一边等。
好？那我们开始吧。Hello, everyone. This is Jing Xiong from China Pharmaceutical University. I am happy to moderate the talk today. Thank you for joining us for the third Chinese American Diabetes Association Happy Hour virtual talk in 2024 to 2025 season. We are happy to have 2024 CADA Achievement Award winner Dr. Yong Liu from Wuhan University as our speaker today. Dr. Liu received his bachelor and master degrees in biochemistry from Peking University and obtained his PhD degree in microbiology and molecular genetics from Rutgers University. After his postdoctoral training at the University of California, Santa Barbara from 1995 to 2000, Dr. Liu worked as a senior scientist at the immunology section of Alicure Mankind Corporation, a California-based biotechnology company. In 2003, Dr. Liu joined the Institute of Nutritional Sciences as a principal investigator at SIBS, Chinese Academy of Sciences in Shanghai. In 2015, he joined the College of Life Sciences at Wuhan University. Dr. Liu has received a number of awards, including the Distinguished Young Scientist Award from the National Natural Science Foundation of China in 2009. He has served as the chief scientific investigator for 2 national research programs from the Ministry of Science and Technology of China. Over the last 20 years, Dr. Liu's research has also been supported by numerous grants from the National Natural Science Foundation of China. So he's running a well-funded collaborative research group. Dr. Liu has made a lot of achievements on molecular and cellular mechanisms that link ER stress to the pathogenesis of metabolic and inflammatory diseases. He's not only a pioneer in research, but also a good mentor for a lot of young investigators. A lot of them I know. Most of them have been independent researchers in China. Today, he will share his insights on the ER stress signaling in metabolic control and stress management. We also have 4 panelists today who are outstanding scholars interested in the field of ER stress, which are Dr. Lin Yang from University of Iowa, Dr. Shen Yisun from University of Virginia, Dr. Yinfeng Deng from City of Hope, and Dr. Ye Weiji from Chongqing Medical University. Welcome. Now, let's pass the stage to Dr. Yong Liu and enjoy his insightful talk today. Please take over. Thank you.
Okay, thank you so much, Jing, for the kind introduction. I'm so happy to be here back at the webinar series with the CADA Happy Hour seminar. CADA is always a family, although across the Pacific Ocean, we can still have this activity. This is very nice. Today I'm going to talk about, share with you some of the recent work from my lab. That's ER stress signaling in metabolism and stress control. We all know that in eukaryotic cells, the ER is a very important organelle. Actually, it's the largest organelle. It's a network of intracellular membranes. It performs many critical functions. It's an active storage site for intracellular calcium. And also it's the site of for protein folding, modification, and transport, especially for membrane-bound proteins or secreted proteins. Also, the ER is the place for lipid biosynthesis, for example, for the lipid droplet biogenesis. ER is also known to have nutrient sensing functions. It's involved in many signal transduction events. Recent cell biology field has focused on the organelle interactions. Actually, ER can interact with many other organelles, including mitochondrial, lysosome, endosome, lipid droplet, almost every other organelle. So what's ER stress? Initially, an increased accumulation of unfolded or misfolded protein would cause the ER stress state. Under these conditions, the cell would mount a UPR response called unfolded protein response. So ER stress either is adaptive or is maladaptive. The maladaptive ER stress has been implicated in many diseases, including obesity, insulin resistance, and type 2 diabetes. There are three canonical signal transductions events that govern the UPR. They are mediated by three ER resident transmembrane proteins, ATF6, LRU1, and PERK. ATF6 is a transcription factor localized at the ER, is activating transcription factor 6. Upon activation by ER stress, ATF6 would translocalize to Golgi apparatus that cleaved by S1, S2P proteases and generating a cleaved active transcription factor ATF6. PERK is a pancreatic ER kinase. Upon activation, it dimerizes and phosphorylates the EIF2 alpha at serine 51. This would shut down the cellular protein translation. Meanwhile, it would activate the ATF6, ATF4 transcription factor through translation control. This would go to the nucleus and activate the downstream UPR genes. LRU1 is a very interesting and fascinating molecule to me. It contains a luminal domain sensing the unfolded protein or misfolded protein in the ER lumen. The cytoplasmic portion contains two enzyme activities. It's a serine threonine protein kinase. At the C terminus is RNase, is an endoribonuclease activity. Upon activation, LRU1 is known to dimerize or oligomerize, activating through autophosphorylation and activating the RNase activity. One traditional best characterized substrate for LRU1 RNase activity is the mRNA encoding XBP1, the Xbox binding protein 1. LRU1 can cleave two sites and supplies out a small intron in mammalian cells. In human, is 26 nucleotides. After cleavage, the RNA gets ligated by an RNA ligase. Then that's a conventional RNA splicing event catalyzed by LRU1 and the RNA ligase. This would generate a spliced form XBP1. So the three UPR pathways coordinate and collaborate with each other to drive the UPR gene expression program, including ER biogenesis, ERAD pathway genes, as well as many metabolic enzymes. LRU1 shortened for inositol requiring enzyme 1 is an ancient stress sensor. Originally, it was identified in yeast by Kuzutoshi Mori and Peter Waters' lab. Over the 30 years, it turns out that the mammalian LRU1 has two genes, two isoforms, LRU1 alpha and beta. LRU1 alpha is ubiquitously expressed, with the beta expressed in the gut intestinal epithelial cells. Many studies indicate that mammalian LRU1 alpha performs many functions under stress conditions. As I mentioned earlier, LRU1 possesses dual enzyme activities. It's a kinase and RNase. Upon activation, it undergoes trans-autophosphorylation and oligomerization. In addition to splicing generation of XBP1 mRNA, it can also degrade the select species of mRNAs. This process is termed regulated IRE1 dependent decay, shortened for RIDD. IRE1 is also known to interact with many other protein partners to form condensates. So IRE1 can have a diversity of signaling regulatory activities. Here shows the structure of the cytoplasmic portion, the kinase and RNase. Actually, there are many phosphorylation sites. Within the activation kinase domain, there are 3 phosphorylserine sites. One of them, serine 724, we've got an antibody to recognize the phosphorylated IRE1 and to measure IRE1 activation state. Over the years, we think IRE1 is actually integrates various stress signals. In addition to sensing unfolded or misfolded proteins, IRE1 can also sense many metabolic signals or inflammatory signals. Like in beta cells, it can be activated by glucose stimulation. In the liver, it can be activated by free fatty acids or glucagon, insulin, or in immune cells, it can be activated by LPS or cytokines. In terms of mechanistic actions, IRE1 can utilize the XBP1 to drive the downstream UPR gene expression programs, as well as RIDD. You can degrade select usually cell type specific RNA substrates, as we find out. Also, IRE1 can form a multi-protein complex. As you can see here, this is the immunostaining of IRE1. We can see in cells, it forms a large foci or clusters. So far, we know IRE1 can interact with the TREV2, IKK beta, or STAT3. It performs many regulatory actions in different signal transduction pathways. My lab has been trying to elucidate how the IRE1 is like a large protein machinery in the cells, how it operates in tissue homeostasis. How IRE1 behaves in cells, particularly the cellular nature of the IRE1 clusters or condensates. Over the years, we have examined the IRE1's metabolic functions in different metabolic tissues. For example, in the POMC neuron, IRE1 promotes obesity through affecting the browning in the wet adipose tissue, actually. We also find IRE1 regulates insulin biosynthesis. It regulates the beta cell proliferation in the state of obesity. In the liver, it can regulate gluconeogenesis, lipogenesis, fatty acid oxidation, ketogenesis, etc. Recently, we find in adipose tissues, myeloid IRE1 can promote metabolic inflammation in the adipose tissue, causing metabolic dysfunction in adipose tissue, leading to obesity and type 2 diabetes. So today I'm going to focus on some of recent studies. That's the area we are interested for a long time, because the tissue-specific RNA substrate regulated by RIDD mechanism is poorly understood. actually it looks like a box of chocolates. We don't know what we are going to get next before we find out what's the downstream substrate for ARE1 RNase activity, particularly in animal models. Also, I'm going to share a little bit recent work on the ARE1 protein machinery in stress control. So for many years, we are interested if the question whether ER stress promotes adipose inflammation and dysfunction in obesity. We focused on the macrophages in adipose tissue. As we know, the M1, M2 polarization at the time is a hot topic at the time. We thought, well, ARE1 maybe control the balance of between M1, M2 polarized macrophages. It turns out using the myeloid knockout ARE1 animal models, we found that overnutrition can really can indeed induce metabolic ER stress in the adipose tissue. During this process, ARE1 is suppressing M2 polarization. However, we don't know whether it's through RADD mechanism. We think we know it's dependent on its RNase activity, but we haven't been able to pin down the molecules regulated by ARE1 RNase at the time. Following that work, we went on to study whether ARE1 regulates different fat cells. As I don't think I need to go to the details about the different white adipose adipocytes in this audience. We know that white fat cells is specialized for storing chemical energy. We also have a brown or beige fat cells that express the high level of UCP1 to drive thermogenesis. So the question is, we want to know if ARE1 regulates the adipocyte functions in different fat cells. Initially, we made the observation. 听得见吗，叶伟？喂？
可以的，可以听见。
可以听得到。刚才有个人可能在说话。
Okay.
给他 unmute 掉了。
So we made the observation that upon cold exposure, we maintain the mice at 8 degrees or 4 degrees for a period of time. Then we isolate the inguinal or brown adipose tissue or epididymal white adipose tissue. We measure the ARE1 phosphorylation levels. As you can see in the inguinal rat and the bat, we can see cold-induced phosphorylation of ARE1 as well as XBP1 spliced form production, indicating the activation of ARE1 XBP1 pathway upon cold exposure in thermogenic fat. But this doesn't happen in epididymal white adipose tissue. Then we thought ARE1 in adipocytes could regulate the browning thermogenic process. Then we generated a adipocyte knockout using adiponectin promoter-driven query and delete ARE1. As you can see here, upon cold exposure, we can see the remodeling of the inguinal white adipose tissue. Once we deleted the ARE1 gene in the adipocytes, you can see an increased or higher level of remodeling of the browning, as indicated by UCP1 immunostaining also. Then we went on to look for what could be the mediator downstream of ARE1 regulating the thermogenic process. Then we through RNA-seq transcriptome analysis of the cold-induced wet adipose tissue, we focus on a bunch of transcription regulators or transcription factors. And we can see upon ARE1 deletion, a bunch of transcription regulators are altered. And one particular gene called PTC1 alpha caught our attention. Everybody knows PTC1 function in the thermogenic mitochondrial metabolism as well as browning. And indeed, we looked for the ARE1 typical ARE1 recognition motif for ARE1 cleavage. We did find the consensus sequence conserved in both the human and the mouse PTC1 alpha mRNA, and they can form the short stem loop structure, which can mediate the efficient cleavage by ARE1. Then we made a RNA substrate, fluorescent substrate using recombinant ARE1 protein to cleave this RNA substrate. You can see ARE1 can indeed degrade PTC1 alpha mRNA. When we mutated the cleavage site from GC to CC, this would abolish ARE1's cleavage activity. Also, we used the RNase inhibitor, Formula 8C, which is a specific inhibitor for ARE1 RNase activity. This would also suppress ARE1's cleavage of PTC1 alpha. This XBP1 mRNA is used as a control. So basically, the model is we think in adipose tissue, ARE1 can respond to cold stimulation or beta-3 agonist activation. ARE1 can be activated by PKA through phosphorylation, and the overactivation of ARE1 during obesity, ARE1 could degrade PTC1 alpha, leading to impairment of the browning process, and then the obesity and the glucose intolerance. And of course, ARE1 also, in addition to RIDD decay of PTC1 alpha, ARE1 can also directly inhibit the lipolysis. This mechanism, at this stage, we are not very clear about the mechanism yet. So the second question is, we are also interested in the vascular endothelium. We want to know if ER stress in endothelial cells could play a role in metabolic regulation and the metabolic control. Because we all know that EC cells perform many important functions in different tissues. And the recent tissue-specific single cell studies have shown that the EC cells possess a very remarkable tissue-specific heterogeneity. Initially, we tackle this question because we all know in the state of obesity in the adipose tissue, the ECs undergo remodeling and dysfunction. We thought ARE1 may play a role in the EC cells in adipose dysfunction. So please get ready for the surprise. To address this question, we generated a tamoxifen-inducible EC knockout to deplete the ARE1 gene in the EC cells. And here is isolated the EC cells. We using immunoblotting, we show the AR1 can be deleted in EC cells after tamoxifen injection. Then we maintain the mice on high-fat feeding and to look at if the AR1 deletion in EC cells would affect obesity adiposity. Apparently, we didn't observe any changes following high-fat feeding that suggesting AR1 in EC cells has not much a role in adiposity regulation. However, when we tested the glucose tolerance using the GTT or ITT, we observed a significantly impaired glucose tolerance following high-fat feeding in the knockout mice. But we didn't observe any alterations using the insulin-regulated glucose disposal. So that obviously we think there must be something happening in the pancreatic islets. Indeed, we measured the serum insulin levels. You can see after high-fat feeding and EC knockout mice showed a lower level of insulin in the blood. And here is the insulin level during the GTT test. In the EC knockout mice, they have a lower glucose stimulated insulin production in the circulation. So pancreatic sectioning analysis, we analyze the islet number or islet mass, and you can see the only after high-fat feeding, the knockout mice showed a lower islet number and a lower average area in the knockout mice. Through immunostaining, we use CD31 to stain the EC cells. And you can see after high-fat feeding, EC knockout mice showed a lower abundance of EC positive signals, suggesting a lower vascularity in the islets in the knockout mice after high-fat feeding. By contrast, we didn't observe significant changes in the inguinal white or epididymal white adipose tissue when you look at the CD31 positive EC cell signals. We didn't see the changes in the EC cell abundance in this adipose tissue. Then later on, we want to know if AR1 regulates important regulator in the EC cells in the islets. This is surprising because we thought AR1 in EC cells could affect other metabolic organs. However, we didn't observe any changes except in the islets. So this is suggests the AR1 in the EC is very important in the islets. Using the islet RNA-seq data from the fed mice, and we analyzed the gene expression programs in the islets. We found there are a lot of insulin secretion pathways are altered after AR1 is deleted in the EC cells. When we analyze the upregulated or downregulated genes, one gene caught our attention, which is TSBS1. Because using the database, there are a lot of people who did the single cell analysis of the islets from the high-fat model, and THBS1 is known to be specifically expressed in the EC cells in the islets. Because in the knockout mice, we found THBS1 is upregulated. And then we confirm this by using RT-PCR analysis of the islets. And we found the ARE1 knockout would result in an increased expression level of THBS1 and the protein level also. So what is THBS1? It encodes the protein called thrombospondin 1. This is a endogenous inhibitor of angiogenesis, and people have shown that the THBS1 in the circulation levels is associated with prediabetes. Also, there are people who did the study showing THBS1 has a very important role in beta cell function regulation. But the exact role of TSP1 in beta cells remain to be dissected. So we thought, well, TSP1 may be a molecular target of ARE1 regulation of the EC function in the islets. Then we use the islet-derived EC cells, EC cell line called MS1 cells. As you can see here, when we knock down ARE1 expression, you can see higher expression level of THBS1 mRNA, and in parallel with the lower XBP1 splicing. And you can see here, upon glucose stimulation, TSP1 is stimulated from MS1 cells, and when we knock down the expression of ARE1, the production of TSP1 is increased. Then we analyze the proliferation of MS1 cells. We showed a knockdown of ARE1 would lead to impairment in MS1 proliferation, cell number, and cell CCK assay also showed the same changes. And this is dependent on the THBS1, because when we double knockdown this, this would reverse the effect of ARE1 deficiency on MS1 cell proliferation. So you see because the ARE1 deletion causes an increase the mRNA level of THBS1, then we thought that it could be another RIDD substrate. And indeed, we found a similar stem loop structure. That's the typical recognition structure for ARE1 cleavage. We also found a consensus cleavage site with the GC site absolutely conserved. We did a similar analysis showing that THBS1 is a bona fide substrate for ARE1 RIDD regulation. And you can see the incubation of the mRNA with the ARE1 causes its decay, and the RNase inhibitor forming ADC could block its degradation. So right now we have a model. We think the ARE1 in EC cells, particularly in the islets, it regulates a very important downstream EC cell function regulator, TSP1. This would cause the changes in the microvascular density, which would support pancreatic adaptation or expansion in the state of dietary obesity, which would important for increase the insulin production in the presence of insulin resistance. As you may noticed, we have never showed any metabolic functions in the muscle. Actually, we are interested in the metabolic function of the muscle. As a matter of fact, Qin Li, Lin Qi, once asked me to send the animals knockout mouse to his lab because he's interested in muscle metabolism. At the time, we got some difficulty to ship the mice to the US, so we did the experiment for him. We didn't find much of phenotype. When we knock out ARE1 in the muscle, we didn't observe any obesity or diabetes phenotypes. Once we have the mice, we thought, well, the muscle is very important for physiology, and we are also interested in ER stress signaling in skeletal muscle, whether it regulates muscle regeneration or does it play a role in muscle loss, particularly in aging-associated conditions, sarcopenia. Once we use the MILD gray, we knock out ARE1, we use the simple model using cardiotoxin-induced muscle damage. And you can see ARE1 deletion caused a more severe damage and a slow, delayed regeneration process once you quantitate the muscle cell myofiber. In collaboration with the Zhengji's lab, we used the MDX genetic model and deleted ARE1 in muscle cells. And we also observed a similar phenotype. That's the ARE1 deletion caused the more severe muscle damage, muscle loss. Mechanistically, it's the same thing. We made a wild guess. We thought ARE1 may regulate some protein regulators. We gambled. We thought it could be regulating something like myostatin. And indeed, when we look at the mRNA sequence, we found the classical typical ARE1 cleavage site in human and mouse myostatin mRNA. And indeed, we did a similar RNA decay assay showing ARE1 during damage-induced stress. ARE1 can decay myostatin mRNA to promote muscle regeneration. So we had the muscle knockout mice, and the COVID-19 came. We had to maintain the mice in the cages and got them old. By chance, we look at the older mice. We made another observation accidentally. We didn't intentionally to do these studies, but one interesting phenotype is once the mice get older, you can see increased muscle loss in the muscle ARE1 knockout group. And also the muscle function in terms of grip strength and the hanging time, the older mice showed a decline in their muscle function in the aged mice. Then we measure the muscle weight. As you can see, the different muscle weight was decreased in the aged mice following in the ARE1 muscle knockout mice. They showed a higher muscle loss. But in the adipose tissue, wet adipose tissue had no significant changes. HE staining also showed the impaired regeneration in the muscle and the smaller myofiber myotubes in the aged knockout mice. So this suggests muscle ARE1 deficiency would accelerate the muscle loss in the aged mice. So what could be the substrate, the mechanism? Then we did RNA-seq again, and we analyzed the gene expression programs influenced by AR1 deficiency in muscle cells, in muscle tissues. And one pathway pops out. And this is all related to metabolism of prostaglandin. And one gene is most dramatically altered upon AR1 deletion in the aged mice. And you can see here, AR1 knockout mice shows a very high level expression of a gene called HPDG. HPDG encodes the enzyme that degrades prostaglandin, which is a very important molecule in muscle aging regulation. Because a couple of years ago, Helen Blau's lab has shown that this 15 PGDH is very important in muscle aging, because in aged mice, this enzyme goes up and would lead to decreased PGE2 production, which drives the muscle loss process, impairs almost every aspects of muscle function. So we thought, well, this is comes together. ARE1 may also regulate HPGD through RIDD, because the ARE1 knockout mice shows a very high level expression of HPGD. However, we did many experiments. We didn't observe any RNA decay phenomenon. So we thought, well, this could be the other way, could be the transcription control. ARE1 is acting in a different fashion. And indeed, when we use the promoter reporter of HPGD expression, and you can see overexpression, co-expression of ARE1 would lead to decreased promoter activity. ARE1 knockout, you would see a higher promoter activity, suggesting ARE1 is a suppressor of HPGD transcription. Then we look at the promoter and found many AP1 sites. And in the aged muscle tissues, we see the knockout muscle showed a very higher expression level of c-Jun, but not in C-Force. So right now we are trying to work out the details. We think ARE1 regulates c-Jun protein stability through proteasomal degradation, or its ubiquitination. So right now we are trying to work out all the details. This data shows ARE1 is a very important regulator. It protects mice from age-associated skeletal muscle atrophy or muscle loss. Now I will switch gear and share some of the recent work on the ARE1 clusters or foci. Because for a very long time, ARE1 is known to form large clusters, usually during ER stress, or when you overexpress ARE1, you can easily see ARE1 foci. I thought, well, ARE1 foci may not be ARE1 itself, or it's just a polymer. It could be a more complex cellular structure, particularly in recent in the recent discoveries about phase separation condensates, protein condensations. We thought it could be something related to stress granules. So we did some initial experiments, and we used G3BP1 is a stress granule co-structure protein. And indeed, you can see ARE1 can form clusters upon TG treatment, and remarkably, you could see the very nicely co-localize the ARE1 foci with the stress granules. And this happens not only in response to ER stress, but also to oxidative stress in cells treated with arsenite, as well as heat shock stress in cells incubated at 42 degrees. So this is not restricted to ER stress. It's a general stress response, suggesting ARE1 clusters has something to do with the stress granule. Then we confirm this using a system called a dimerization-dependent fluorescent protein reporter. And basically, we made the fusion proteins. And when the two proteins associated with each other would gives out the bright light, fluorescent signals. And here you can see ARE1 is green. And the two stress granule markers, G3BP1 and the T1A1. And when they interact, it's red condensates, and ARE1 is the green condensates. Then you can see upon TG treatment, the ER stress-induced formation of ARE1 clustering with the stress granule. The different observations. In addition, we also see the ARE1 stress granule classes can undergo both fission and fusion using the DDFP monitoring system. So what drives this condensates formation? We look at the ARE1 structure. You can see that's a linker region linking the transmembrane domain and the kinase RNase domain. And for a long time, I've noticed this in the linker region. ARE1 sequence contains a lot of serines, as well as a bunch of glutamines. These are typical segments in the proteins containing intrinsically disordered regions. We know that ADRs are very important to drive protein condensation phase separation. So we thought this could be very important for ARE1 clustering with the stress granules. So we employed another system called the light-inducible optodroplet system to monitor the ARE1 phase transition. And this basically use the protein called Cry2. Upon blue light activation, Cry2 would form a dimer. This would drive protein phase separation from clusters. And you can see here, when we using system, you can see the ARE1 upon light, ARE1 can form these large clusters following light activation. In contrast, when we delete the linker region, we don't see this apparent cluster formation any longer. That means the linker mediates ARE1 phase transition. Not surprisingly, when we delete the linker region, the ARE1 could no longer form the co-clusters with the G3BP signals of the stress granules. Then we thought, well, either ARE1 could influence the stress granule formation, or stress granule would influence the ARE1 clustering. And we didn't observe any effect of ARE1 knockout on the stress granule assembly. So here you can see when we inhibit or block stress granule formation with the cyclohexamide, we no longer see the formation of ARE1 clustering, suggesting the here also when we double knock out the G3BP1 and 2, this stress granule formation got impaired. We can no longer see ARE1 clustering foci formation, suggesting stress granule from assembly is a prerequisite for ARE1 clustering to happen. So what's the what could be the potential function? Because we know ARE1 activation leads to XBP1 splicing, and this pathway utilizes the RNA ligase. This RNA ligase is called the RTCB, and we overexpress EGFP fused RTCB and try to see if this this RNA ligase is also colocalized in the ARE1 cluster and SG condensation complex. And indeed, you can see upon TG stimulation, RTCB is a complex together with the ARE1 and the G3BP signals, suggesting they form a very large protein complex. And in addition, we also see an enriched level of the substrate XBP1 mRNA, and when we knock out the G3BP to disrupt stress granule formation, we see a lower level of XBP1 mRNA substrate. So that suggests the ARE1 X stress granule condensation forms a working platform for more efficient ARE1 splicing activity for XBP1 mRNA. To test this, we engineered artificial protein using the kinase RNase domain and using the fused with the NTF2L portion of G3BP. So this would deliver the kinase RNase domain to stress granules, independent of ARE1 or the ER membrane. And you can see here, this artificial fusion protein can localize to the stress granule following TG's induced ER stress. And when we isolated these cells, localize the kinase RNase protein. So the working model is, we think in response to external stresses, different stresses, ARE1 dimerization or oligomerization, it can condense with the coalesce with the stress granule through phase separation process. And this would generate a very efficient work platform for XBP1 because it enriches all the biochemical components of the XBP1 splicing pathway. This would allow cells to cope with the stress condition for survival. So still, we don't know the exactly ARE1 would regulate the other properties or functions of stress granules. Also, we don't know if this mechanism is involved in ARE1's metabolic control functions. So we are going to tackle a lot of questions to be addressed down the road. Finally, we want to know how ARE1 works because people have considered ARE1 as a therapeutic target in the drug companies, and I think we have to know how ARE1 operates to know the detailed molecular mechanisms in different disease contexts before we can more efficiently or precisely target ARE1 for therapeutic agent development. We want to provide more clear clues or basis for that endeavor in the future. So finally, I want to thank my graduate students and the former graduate students and the students, current students in the lab, as well as many friends and collaborators in the audience. Thank you so much. I hope I can answer some of the questions you may have. Thank you.
Thank you, Dr. Liu, for the wonderful talk. And such a big group, and you can make all these people get full funded. And I'm generous.
Thank
and so many interesting stories. And I'm just curious, is that the kindness activity of IRE1 same is regulated same in the same way with this RNase activity?
That's a good question. Usually people think the kinase through autophosphorylation that would cause the conformational changes, which in turn activate the RNase domain to open up a docking site for the RNA substrate. I thought the kinase has other functions, but we don't have very convincing data yet.
Yeah, it looks like the kinase domain is nearer than to the linker site, right?
Yeah, it's the disordered linker, then followed by kinase and then RNase domain. The bridge between kinase and the RNase region is also very important, as Qilin's group has shown many years ago. In addition to autophosphorylation, other kinases can also phosphorylate the ARE1. There are a few examples now. We show the pKa can phosphorylate the ARE1, and Chen Xi's group has shown ERK can phosphorylate the ARE1. So the phosphorylation sites are very difficult to identify or precisely measure. That's a very we know it's important, but we don't know which site is doing what. So it's very complicated.
Yeah,
there are many phosphorylation sites. Yes.
And you still have many open questions to work on.
Yeah,
Okay,
it's
I will let the panelists to ask questions. So
Sure.
please, the first one, Dr. Lin Yang from University of Iowa.
Dr. Liu, excellent talk.
Lin.
Yeah, hi. From physiological to pathological relevance and also to cell biology, very, broad aspect and a very insightful talk. My first question it is, which isoform is enriched in the endothelium cells in terms of IRE1? Is alpha or does it has beta isoform in, in endothelium cells?
We thought about it. We never paid attention to beta. I think we should have started to analyze more precisely which cells, which tissues, the alpha beta expression pattern. The old one, I don't think they are very accurate or sensitive.
Yeah, because your
knowledge.
alpha, your EC cells alpha knockout in general doesn't have a very profound phenotype, but there are probably other beta form I don't know. So it's not a very
Right.
yeah.
I asked my students. They looked through the single cell data from the database, the published database. We didn't find much of beta, I think.
So I just I'm curious too. So I don't know. We always emphasize alpha.
Right.
It is a beta. second question regarding the excellent talk in the excellent study in the brown adipose tissue RE1 function in terms of rate action to process speech PGC1. So I'm wondering, do you think it's possible because the brown adipose tissues the activation is not sustained, it's dynamic. You activate thermogenic action, but it could go back. So it's a maintenance of brown fat, from the highly active mitochondria enriched stage, go back to to the silence or basal condition. Do you think the RE1 mediated PGC1 degradation? For long shot, probably it can help for maintenance of brown adipose tissue.
Yeah, how
Yeah.
physiologically how important? I think it's feedback
Could be
control.
adaptive feedback. Because
But
when you do the CL treatment or cold exposure, how long, I don't know how long you treat them or how long
cold
you
exposure is that
yeah,
we did 3 days to 2 weeks, I think.
2 weeks.
Yeah.
I'm just curious. Yeah, I would,
This
excellent.
This happens in the beige
In the
adipose,
beige cells, yeah.
not a brown adipocytes. We tried so hard, it didn't happen in brown adipose tissue at all.
Okay.
We had a hard time dealing with the reviewers' questions actually to tell why it didn't happen in the brown adipocytes.
Oh, got you.
It didn't happen. And we used the UCP1 query that got us a lot of questions from that model. I didn't have time to go all the different models here. But it's all published in there in the supplemental data.
All right, I will pass to the next panelist. Yeah. Thank you so
I follow up the EC stories a little bit. So you just mentioned the only phenotype is related to the pancreatic islet, right? The other part you didn't see any phenotype. Do you think is that there's no target for the IRE1 in other part of the EC cells, or just you need a different insult?
Exactly, I think you are right. We just use the high-fat feeding people in the lab. Usually, that's a favorite model. I don't think that's the best model to tell you the truth. I think we do have another injury. I'm not sure what model it is. I think we do have other phenotypes in other tissues, not just the islets.
So the conversion can still be open.
Right.
Okay, so we
Can
have
I ask
the
a question?
second. Yeah, sure.
Young, can I ask you a
Happy to
question?
see you.
Rolling.
Hey, really great seeing you, man. And I apologize. I actually forgot about what I asked you about the muscle.
A long time. When you were in Cornell.
Yeah.
We talk about the shaping the mice, but we thought you told me it's a hassle. Forget about it.
Right. So I have a question about this stress granule. I think this is fascinating. The stress granule. How did all the things are coming together with the membrane, right? The IUF is membrane bound, and then this XBP1 splicing enzyme. It's also, somehow all clustered together. Like,
right.
I'm just having a hard time imagining this, because these are ER membrane bound. Do you see a ER membrane? Do you see this thing is attached to the ER membrane, this stress granule?
We did that actually. JR's lab has shown stress granules P bodies. They are actually tethered to the ER.
Tethered to the membranes.
To the ER membranes. The stress granules are cytoplasmic.
Right.
They usually happen around the ER membrane.
Right, but
they thought that's an anchor protein. I thought the RE1 is the anchorage protein.
But in your case, it has to be really close, right? It has to be really close to the membrane.
Very close. We actually use the dimerization using a ER protein marker and the stress granule marker. You can see the fluorescent signal suggesting their tethering or contact sites.
Do you see this in vivo or in pancreas that the SNS cells where they have tons of IRU alpha and they're constantly making proteins. Do you see the IRU alpha form these foci?
No, we didn't try the islets.
No, I mean the SNS cells, the exocrine part, the one that the exocrine actually has tons of IRU alpha, has really high level of IRU alpha.
We never tried to stain. I don't think the antibody is good enough for staining IRU1 in tissues.
Okay.
Very hard.
Right. Great seeing you, and sorry to miss the party next week.
I thought you could be the panelist. I thought you are too busy. Good to see you.
Can't miss your seminar. Yes.
Thank you for showing here, Dr. Qilin. And next we have our panelist, Shenyi.
Hi, Dr. Liu. Really enjoy your talk. Thank you for sharing those exciting studies. I have a question about the activation of IRU1 alpha, for example, during angiogenesis, and for example, during the aging of muscle. Do you think IRU1 alpha, for example, the IIDD activity or other kinase activities is constitutively active, or is it induced under stimulation? And if you can comment on that.
I think that's the most puzzling part of the field. How IRU1 is activated. Well, the membrane, ER membrane alteration, they call the membrane stress, bilayer stress, could activate the IRU1. I don't know why cytokines can also induce IRU1 phosphorylation, splicing activation. You can always go through seeing protein synthesis or protein folding load, but that may not be all the situation is protein folding. I don't know. I think other factors that modulate IRU1, like modifications or phosphorylation, could trigger its activation cluster. People are starting to see IRU1 activation happens in the subdomains. That means IRU1 concentration is very important in the local compartment, I think. But we don't have much evidence showing that precisely that's happening. That's the case. I don't know. It's a good question. I think you may know better than me. I don't know.
Thank you very much. It's great seeing you again.
Good to see you.
Okay, so we have third panelist, Yinfeng Deng, Dr. Yinfeng Deng from City of Hope.
Oh, hi, thank you, Jing. And it's great talk, Dr. Liu, and it's so honored to be here. Yes, I couldn't imagine how fascinating the IRU1 alpha can do multiple jobs. The stress granule part obviously is very unexpected to me, and I look forward to hearing more progress. I have a question. I just want to see how you think about it. The clustered IRU1 alpha versus maybe not that clustered, still spread out at the ER membrane. You think their target genes could be different? because now we can actually design experiment to test the target genes in 2 different settings. Have you ever thought about that, or have you tried anything?
Frankly, I think the stress granule clusters, when you see it, they are so large. They are very big. I'm sure there are very tiny, small ones that are operative. That's also activate. I think Peter Walters' group showed that the trimer of RE1 is actually active form. It can catalyze the splicing. It doesn't need to be a oligomer or a foci, I think. It's still an open question. We don't know when it goes to precisely splicing XBP1 cleavage, just the 2 sites. And the decay, RIDD decay, we don't know how that happens actually. Just a cleavage once, followed by exonucleases or other players. We don't know anything about that RIDD process. I think it takes a complex of proteins, but I don't have much approach to address that yet. Maybe we can collaborate. You have ideas. Thank you for that's a tough question. I don't know really.
Well, I believe you will see and show a lot of interesting things in the following years. Thank you, Dr. Liu.
Thanks.
Okay, so next panelist, Dr. Ye Weiji from Chongqing Medical University.
Hi, Professor Liu. Hi.
Hi.
It's great talk, and I'm sorry for you that you will give a talk tomorrow morning in Hangzhou. I know that.
Yes.
So, but I really enjoyed the talk. So my question will be, the IURS and IDD, you have so beautifully demonstrated the substrate and also show the sequence, like UGCA, those things. Do you think that sequence also somehow with hairpin-like structure make it enough or substrate for IO alpha, or still you still need to confirm? I mean, you show that sequence, right? The dependence of the sequence mRNA with hairpin-like structure. So is that structure and that sequence to be a substrate? And originate.
Yeah, I know. That's a question always on my mind also. People just read the papers. They show that this stem loop is RE1 cleavage. That's a site. But when people do the in vitro analysis, that's a trap. I think you incubate RE1 RNAs, recombinant purified protein with any mRNA. RE1 can degrade it. If you run the gel, you can see a lot of letters. People use that for insulin, right? In the cell paper. But in vivo, in cells, we don't know the decay. Probably the stem loop would trigger the decay process, even give it a kickoff signal with the cleavage. That's what I speculate. I don't know. That would be the real case. Also, there's a guy, AV, in Genentech, they proposed the riddle sequence. So RE1 can cleave mRNAs even without stem loop. Then the cleavage still happens, but we don't know. Probably the GC site is well conserved. I don't think that's a very very unique structure feature that's required for cleavage. I don't think so.
Also, I think I have a question.